everyone, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society for Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. Today's talk is the 18th Plantae Presents webinar in this series. If you haven't already done so, check out our archive to watch all of the previous talks in this series. You can find information on our blog and subscribe to our YouTube channel as well. If you're having trouble connecting or need to leave the webinar early, know that a recording of this webinar will also be made available along with associated materials within a few days. Today's talks will each be 20 minutes, followed by 10 minutes for questions. If you have questions for our panelists, please let us know using the Zoom question and answer Q&A section. You'll notice there is also a chat area open. This area is for you to introduce yourself, say where you're from, and connect with your colleagues. But questions that are specifically for our panelists should be shared within the Q&A section to make them easier for our moderator to navigate. Dr. Jill Eschel is here with us today to moderate this session. Jill is a postdoctoral associate at New York University in the lab of Gloria Caruzzi, where he uses evolutionary and systems biology approaches to study the adaptation of desert plants to dry and nutrient-poor soils. His research goals are to understand how plants evolved and adapt to extreme environments, and to identify genetic components that can be used for improving crop resilience. He is also interested in developing new computational tools and approaches to mine large genomic data to gain evolutionary and functional insights. I'm going to turn it over now to Jill to introduce our first speaker and moderate today's session.
Thank you very much, Katie. It is my great pleasure to introduce today's webinar speakers. Our first speaker today is Elizabeth Vierling. Elizabeth is a distinguished professor at the University of Massachusetts Amherst. She received her PhD in biology at the University of Chicago, studying biogenesis of photosystem 1. She then moved to the lab of Joe Key at the University of Georgia for postdoc research, where she started working on Hitchcock proteins in plants. She then got a faculty position at the University of Arizona, where she dedicated her career studying Hitchcock protein molecular chaperones and the role in thermotolerance. Since 2011, she is at the University of Massachusetts Amherst, where her work focused on the structure and function of molecular chaperones and the mechanism of plant response to environmental stress. She also studied how nitric oxide homeostasis impact growth and fertility and the control of mitochondrial gene expression and respiration. Elizabeth's title for today is From Chaperones to Mitochondria, Surviving Stress. Elizabeth, the stage is yours.
All right, well, I would like to start by thanking Mary and thanking ASPB for organizing this opportunity for me to share my work with you, and also for all the other speakers that have contributed to what is a really wonderful opportunity during this strange time we're all living in. So the story that I want to tell you today concerns the genetic dissection of heat acclimation that leads us to talking about chaperones and ultimately to the control of mitochondrial reactive oxygen species production and intracellular communication. So I think you all know that plants experience a range of temperatures, even on a daily basis. And this is something that has interested my lab for many years. And this is just a graph to illustrate that fact that, for example, looking at maize in a field in Nebraska, the plants can go from the moderate temperature of 25 degrees all the way up to 45 degrees over the course of a few hours in a day. I've seen the same thing measuring eucalyptus leaf temperature in West Sydney, Australia. So plants can experience this dramatic daily temperature change. And we think that this is a common occurrence and that plants are able to acclimate over the course of this daily temperature change. The importance of this process, or the importance of how plants, the temperature rises only gradually during the day, is illustrated in our experimental system, which is Arabidopsis. So I just show here that plants can acclimate to otherwise lethal temperatures. So if you put an Arabidopsis plant at 45 degrees abruptly, and then look at it 5 days later, it's dead. However, if you allow the plant to gradually increase to that temperature, just like you saw in the previous slide for what's happening in the field, the plants survive. So we've been sort of fascinated as to what's happening in this really short time frame during a day that makes the difference between life and death for a plant. So my lab has spent a number of years using genetic techniques to look at how plants do this acclimation process and why is that important. So what we've really focused on then is defining functions required for high temperature tolerance, and have used forward genetics in Arabidopsis to do that by isolating loss of function mutants that fail to be able to acquire this tolerance to high temperature. And this has resulted in the isolation of a number of extremely interesting mutants. So from this genetic screen, we've come up with the following conclusions regarding what kinds of things are important for this acclimation process. So one is protein homeostasis. So it was very satisfying that one of the first mutants that we obtained from our screen was a mutation in the cytosolic chaperone HSP101. So this told us protein homeostasis clearly is interesting and important for this acclimation process and survival of high temperature stress. A second mutant is involved in control of reactive nitrogen species. And so this enzyme S-nitrosoglutathione reductase allows plants to modulate the amount of nitric oxide and the reactive nitrogen species, which when they get out of control are very detrimental to plants under stress conditions. A third mutant was involved in the protection of translation, and this we called HOT3, and this is a mutation in EIF5B, a translation initiation factor. So all of these processes are clearly very important for acclimation to high temperature, and no doubt for processes related to other types of temperature stress. So what I want to focus on today, though, is really this first process of protein homeostasis, and how studying this molecular chaperone HOT1, or HSP101, has led us to our interest in mitochondria and communication within the cell. All right, so protein homeostasis can also be considered to be protein quality control, and this is how any cell regulates the number and activity of proteins in the cell. So HOT1, the cytosolic chaperone, HSP101, is very important in this process. So how does HSP101 actually work? Well, a number of studies, both in my lab, but also many molecular studies by other groups, including Bernd Buchau's group and Susan Lindquist, have shown that HSP101, or CLIP-B as it's known in bacteria, are ATP-dependent disaggregating machines. So you can see a model for this process here, but if you have a native protein, which is denatured by stress, can form protein aggregates, and HSP101, which is a large hexameric ATPase machine, is able to take those proteins and unfold them with the help of other chaperones and eventually refold them such that they are now active again. So one of the questions we wanted to ask was what kinds of other proteins are involved in this process? What are some of the partners of HSP-101? So one thing that we were able to show is that in addition to this chaperone working in the unfolding process of denatured proteins, that also the small heat shock proteins cooperate with HSP-101 to disaggregate proteins. And so that can be seen in this model. Again, we have HSP-101, which is a disaggregation machine. And the small heat shock proteins represented here by the structure of a dodecameric small heat shock protein from wheat. These proteins are in a dynamic equilibrium with their dimeric substructure. And it's this dimer that interacts with unfolding proteins to form larger, more soluble aggregates that are readily unfolded by the HSP-100 machine. So these are studies or this model is supported by both genetic and biochemical studies that have been done by a variety of people in my lab, primarily Iman Basha, Ung Lee, and Min-Soo Kim, and Fiona McLaughlin. So we are very interested in understanding what proteins are protected, and that's an area that I'm not going to talk about today. What I do want to consider is the other aspects that we've looked at with regard to HSP-101 and chaperone function. So these studies have told us that protein refolding and quality control are essential for acclimation to high temperature stress. But are there other HSP-101 partners? Are there other processes involved in heat tolerance? And so this is where we, again, use genetics to get further into this process. So we screen for suppressors of a dominant negative HSP-101 mutant. And we've called these mutants SHOT mutants, or suppressors of HOT1.
Hi, Elizabeth. Real quick before you continue, it looks like we have a gray box showing on our end. Do you have maybe a part of like a Zoom meeting or something over there that you could move to the side? Perfect.
Is that okay?
Yeah, that's much better. Thank you.
Oh, have I been blocking the whole thing?
Oh, no, not the whole thing, just like a little bit of the side.
Okay.
Just makes it easier to see, so.
Okay, sorry about that.
No worries.
Okay, so let's just walk back here for a second. So our genetic studies, then, we were screening for suppressors of the dominant negative of HSP-101, and we found these SHOT suppressors, and I want to talk about one of these suppressors. So one of the suppressors, the first one, not surprisingly, we called it SHOT1, and this is work that's published in the plant cell in 2012. So SHOT1 is a suppressor that bypasses the need for HSP-101. So what is SHOT1? So it turns out that SHOT1 is a mitochondrial transcription termination factor related protein. These are proteins that have a variety of mTORF motifs. These are degenerate motifs of about 30 amino acids. SHOT1 is also thought of as, or named mTORF 18. These proteins, these mTORF proteins, are involved in mitochondrial and plastid gene expression. The SHOT1 mTORF 18 is one of 35 mTORFs in Arabidopsis. These proteins are also found in mammals. In humans, there's only 4 of these proteins. And SHOT1 mTORF 18 is mitochondrial. So this brings us to the question of, let's go ahead and say, how does, or show you that SHOT1 survives heat stress better than wild type and is epistatic to other stress sensitive mutations. And so you see that here Here's SHOT1 surviving heat stress, much better than wild type. Here you can see that it suppresses the HSP101 mutant, but we also found it suppresses other heat-sensitive mutants such as UVH6, which is involved in DNA repair. So somehow SHOT1 is able to overcome the heat stress effects that cause damage to wild type and also to HSP101. So we wanted to ask the question, how can this mitochondria targeted protein partially relieve the need for cytosolic protein refolding by HSP101? And why is it more heat tolerant? So it turns out that after a lot of work by Min-Soo Kim in the lab, our main conclusion is that the reason that SHOT1 is more heat tolerant is that it has reduced oxidative stress. And you can see that here with the raw stain, the wild type showing a large amount of reactive oxygen species in the absence of any of staining in SHOT1. So SHOT1 mutants also, we were able to show, have reduced lipid and protein oxidation, and they also show resistance to paraquat, an oxidative stress agent. So I want to emphasize then that what we're looking at with these mutants, with the HOT1 mutant and the SHOT1 mutant, is we're looking at two major impacts of heat stress. There's protein denaturation and aggregation, and there's ROS damage, and these overlap, no doubt, in protein oxidation. So what we see with HSP101 mutants, they're unable to repair protein aggregated proteins, and they suffer significantly and can't survive the high temperature stress. However, in the SHOT1 mutant, where we eliminate ROS damage, now plants are able to survive even though they can't repair protein denaturation with HSP101. So HSP101 is required for repairing protein damage, but when we reduce ROS damage, plants are able to survive. At this point, we don't know how much of heat stress damage to proteins is due to ROS damage, but we presume that these genetic data show us that protein oxidation is an important component of heat stress damage. Okay, so this leads us to the question, all right, why does SHOT1 and the mTORV18 mutant produce less ROS? So to tell you about that, I need to remind you about OXPHOS in plants and ROS production. You should all be familiar with the mitochondrial electron transport chain, schematically illustrated here, and also that plants have non-proton pumping alternative electron transport components, shown here in green, that allow the electrons to bypass a majority of the electron transport chain and the proton pumping and areas that produce reactive oxygen species. So in order to understand why SHOT1 plants produce less reactive oxygen species, we went ahead and did proteomics on SHOT1 mitochondria. And the mitochondrial proteome of SHOT1 was very informative. We saw increased ribosomal proteins, PPR proteins, alternative electron transport components, molecular chaperones and proteases, and not surprisingly, we saw decreased OXPHOS proteins. So this suggests already a decrease in OXPHOS proteins is that we have reduced activity of oxidative phosphorylation, and that would give us reduced ROS. The alternative electron transport increase is going to also give us reduced ROS, and in fact, alternative oxidase is upregulated 20-fold. And then interestingly, this increase in mitochondrial molecular chaperones indicates that the mitochondria are primed for repair. So this proteomics analysis is also supported by activity studies so that the mutants have reduced complex 1 activity and complex 4 activity, which is shown here by activity stains on blue native gels. So here we have the SHOT1 mutants have reduced complex 1 activity and reduced complex 4 activity. But then related to the fact that the mTOR proteins are believed to be involved in mitochondrial gene expression, if we look more specifically at the mitochondrial proteome, at those genes that are encoded by the mitochondria, we find again that all of the mitochondrially encoded genes are greatly reduced. The proteins are greatly reduced for all of those complexes. In contrast, some other proteins are increased. So this again supporting the fact that this gene, SHOT1, is involved in mitochondrial gene expression, as well as these results being consistent with the reduced production of reactive oxygen species. So then the final thing that I want to address is what is the molecular mechanism of SHOT1 and TRF18? It's clearly involved in some way in preventing the expression of a whole series of mitochondrial encoded proteins that are membrane embedded components of the oxidative phosphorylation machinery. So it turns out that mTORF proteins are one of a family of alpha helical nucleic acid or protein binding proteins. And you can see a variety of these proteins illustrated here: PPR proteins, HAP proteins, OPR, TPR proteins, or the mTORF proteins. And this is the structure of one of the mammalian proteins. All of the plant proteins are predicted to have this same structure. So this begs the question, what does SHOT1 mTORF18 interact with? And it turns out that Min-Soo Kim was able to show that SHOT1 mTORF18 interacts with mitochondrial DNA and with mitochondrial nucleoids. So we find that it weakly binds mitochondrial DNA with some sequence preference, and it localizes to mitochondrial nucleoids. And I really love these pictures. We have mitochondria labeled with MitoTracker, and if you label mitochondrial DNA with Pico Green, you see that it's compacted into nucleoids, as is all DNA compacted in some way to fit inside a cell. And when we label SHOT1 with GFP, we see that it localizes to these same types of structures in the mitochondria. Furthermore, if we look at the SHOT1 mutant nucleoids, they're aberrant, typically sort of exploded out and no longer punctate. So clearly there's some aspect or some importance of interaction of this protein with nucleoids that's resulting in problems in mitochondrial gene expression and then propagating to reduced reactive oxygen species and heat tolerance. But SHOT1 doesn't only interact with DNA, it also interacts with an unusual class of proteins called ATAD3 proteins, or homologous to human ATAD3 proteins. And these are fascinating proteins that contain a matrix localized mitochondrial ATPase domain, but then span both mitochondrial membranes and have what's believed to be in the cytosol a domain of unknown function. So from structures or from work on mammalian proteins, we know that they span the mitochondrial membranes, they're associated with mitochondrial ER contact sites, they interact with DNA binding proteins, and they're recovered in nucleoid preparations. So this is very consistent with what we see for SHOT1. It's an mitochondrial DNA interacting protein, and it's found in nucleoids. So it's perhaps it's consistent with our also finding that SHOT1 interacts with these ATAD3 proteins. So what are ATAD3 proteins in Arabidopsis? So it turns out that Arabidopsis has 4 homologs of these ATAD proteins and 2 clades, and we've designated them as SHOT1 binding ATPases. And you can see the 2 clades here with 85% identity, and between the clades 61% identity. And again, the internal ATPase domain in the matrix and the external DUF domain with membrane spanning regions. Well, further analysis of these 4 proteins in Arabidopsis has shown that they're all localized to mitochondria. Genetics shows that we need 1 SBA gene from each clade for viability. So these are essential, which is also true in mammals and other metazoans. But more interestingly, if we disrupt a SBA function, the plants have reduced complex 1, disrupted nucleoids, and they're also heat tolerant. So the phenotype of the SBA proteins that interact with SHOT1 essentially mirrors many of the SHOT1 phenotypes. All right, so that brings me to our current thinking of how SBA1 ATAD proteins and SHOT1 are interacting. Again, the mammalian literature supports that these ATAD protein is involved in mitochondrial ER contact sites. The SBAs are interacting with SHOT1 mTORF 18 somewhere in this mitochondrial DNA nucleoid interface, and that's impacting mitochondrial gene expression. So looking at this model, I think we can say that both SBA1 and SHOT1 functions are required for mitochondrial nucleoid organization and for assembly of OXPHOS components. And right now, our hypothesis is that when we disrupt this interaction or we disrupt either of these proteins, we may be activating signals propagated from this mitochondrial ER contact site to the nucleus, contributing to heat tolerance. So then just to summarize everything overall, I've tried to show you that from our genetic analysis, we see that surviving heat stress or acclimating to high temperature stress involves protein homeostasis, this molecular chaperone HSP101, which is a protein disaggregase, that it involves the control of reactive oxygen species and the control of reactive nitrogen species and the ability to recover translation after heat stress. And then it's been studies of the control of reactive oxygen species and this interaction between HSP101 and SHOT1 that's led us to uncover the importance of SBAs in communicating potentially with the rest of the cell and also signaling to the nucleus, resulting in survival of heat stress. So I'd just like to say thanks to the funders that we've had over many years, and give a shout out to Min-Soo Kim, who's performed a bulk of the work that I've shown you here, as well as our collaborator in Germany, Christina Kuhn, who has been instrumental in teaching us about mitochondria and how to do many of the experiments I've shown you here. Again, wanting to acknowledge all the current members of my lab who contribute to the overall great atmosphere in the group, and as well as but are working on many other projects. And then also former postdoc Fiona McLaughlin, who did much of the work interaction of small heat check proteins and HSP101. Thank you.
Okay, thank you very much, Elizabeth, for the great talk. We have already a few questions in the Q&A box. So the first question is about growth phenotype of the SHOT1 mutants in normal growth conditions. So for instance, time of flowering, biomass.
Right, right. Yes, so that's something that I managed to avoid talking about in this talk. So yes, the SHOT1 mutants do grow slowly, no doubt because of their defect in oxidative phosphorylation. However, their slow growth is not sufficient, or slow growth is not sufficient for heat tolerance, nor is just being a mitochondrial mutant sufficient for heat tolerance. We've looked at many other mitochondrial mutants, and many slow growing mitochondrial mutants, and they are not heat tolerant. whether or not we can overcome the slow growth to understand the signaling mechanisms that enhance heat tolerance, or how we can better control reactive oxygen species, remains to be seen.
Okay, we have time for 1 or 2 questions. So another question, excellent talk. Do you think that SHOT1 could play a role in heat shock memory response? So after experiencing already another heat stress,
Mm-hmm.
and have you analyzed this in your studies?
So we haven't specifically looked at any aspects of heat shock memory relative to SHOT1, and I really don't have any evidence that would bear on that question.
Okay. Another question, have you measured the level of peroxide lipid after heat stress? Are the shock mutants lowering these?
The question is with regard specifically to
Peroxide, yeah.
No, we've only looked at, we've only looked at protein carbonylation after heat stress and used the dye to look for reactive oxygen species and I guess MDA reactive species.
Okay, last question. Do you think that SHOT1 could be involved in other stress responses? Could other MTRF have the same effect that SHOT1 does?
Well, that's very interesting, and that I didn't point out, but other mTORF mutants have been shown to have stress tolerance. And interestingly, for example, the mTORF mutant SOLDOP10 was isolated as being resistant to singlet oxygen. So there's that connection, and there's at least 1 or 2 other mTORF mutants that have shown tolerance to salt stress or other types of stress. So again, there may be linkages between this control of mitochondrial gene expression or control of plastid gene expression and signaling to the nucleus to enhance stress tolerance.
Okay, thank you very much, Elizabeth, and thank you the audience for their questions. So now we'll move to our next speaker today, Rodrigo Gutierrez. Rodrigo received his PhD with Pamela Green in biochemistry and molecular biology at Michigan State University in 2003. He then moved to the New York University to do a postdoc research work with Gloria Truzzi. He was a pioneer in plant systems biology in understanding nitrogen regulatory networks using Arabidopsis as a model system. He moved back to Chile, his home country, in 2006 and started a strong research program in plant systems biology at Pontificia Universidad Católica de Chile, where he's currently a full professor. Rodrigo has made a key contribution to understanding the mechanisms of nitrate sensing and developmental responses to changes in nitrogen availability in plants. Since 2010, he's studying plants in the Atacama Desert in Chile, one of the harshest environments on Earth, which is his topic for today. Rodrigo, take it over.
Thank you, Gil, for the kind introduction. Can you all hear me well?
Yep, we sure can. Your slide looks great too.
Okay, great. And I want to thank also Katie and Mary for the invitation to be part of this webinar, the Plantae, everybody at Plantae, and also ASPB and ASPB members for having this opportunity. I think it's great to be able to share our work, even sitting here in Santiago de Chile, and in these uncertain times, and be able to get feedback and discuss some of these results. So as Gil mentioned, today I'm going to talk about kind of a different project from what I'm used to talking in the past, which is our efforts to try to understand how plants manage to cope with extreme conditions in the Atacama Desert. You may know that Atacama is the oldest and driest desert on the planet. And besides the lack of water, there's also very intense radiation. The soils are quite poor, and temperatures oscillations during the day also are quite extreme. And despite all these conditions, there is life in Atacama if you know where to look for. I should say before I go much longer that this project was possible to start and to do over the years in collaboration with Claudia Torre, which is also a professor at the same university in the ecology department, which is an expert in Atacama from the ecological and also paleoecological point of view. So a lot of the work that I'm going to show is been done in collaboration with Claudia. So as I said, despite these very extreme and harsh conditions that push life to the limit, if you know where to look, you find surprising diversity. And here I show some images of different plant species that we can find living and in some cases thriving in Atacama in some of these very, very extreme environments. We have been working on a transect, an altitudinal transect that spans the limit for life, as shown here. Just to orient you, this is South America. We're working here in the north of Chile, where the hydrarchic core of the Atacama Desert is found. And we're working on a transect that's east of the Atacama Salt Lake. And as you go up into the mountains, there is more and more water available. If you go below this point, which is approximately 2,400 meters above sea level, there is just not enough water to sustain plant life. And this is what we call the limits for absolute desert conditions. And in fact, we've completed this transect all the way to the coast here to the Pacific Ocean, and we do not find any more plant life below this altitude. Now, if you go towards the mountains, as you go up, as I said, water increases, but also temperature decreases. And this imposes another limit for life, especially for plant life, which is where temperatures are very cold. And so within these two limits, plants can survive, and we've been monitoring diversity and studying these ecosystems over approximately 10 years now. And here I have in this elevation map shown 22 stations, which are a GPS tag where we go back every year to do some studies. And initially this study started very slowly, but over the years it's picking up pace, and I'll show some of the data that we've generated. And a number of groups, both in Chile and also in other places, most importantly, Gloria Cruz's group, as we are now collaborating with us to try to understand how plants manage to live in these ecosystems. Okay, so just to describe the environment, we have two meteorological stations where we monitor climate parameters as shown here: diurnal temperature, diurnal radiation, soil water content, precipitation, and so on. These are measurements taken every hour, and we have over 3 years of continuous measurements and counting. I would just like to highlight a few of the things that I have already mentioned, but this is a graph of the diurnal radiation. And on average, plants here exhibit or receive 3 more times than the average radiation in the planet. So this is a place with extreme solar radiation, particularly UV radiation. Also, in addition to have very limited amount of water, the water is also, or the precipitation occurs in very specific events, mostly in what's called here the summer winter, which is produced by the monsoon from the effect from the monsoon from the Amazons that heats the Andes Mountains. There's precipitation in the mountains, and so plants receive just a few days of precipitation, and then the rest of the year is pretty much gone. So in addition to the climate, we have characterized the soil substrate where plants live. We've done elemental composition analysis, and this is all mostly sand soils with about 80% of sand. And at the elemental composition level, throughout this transect from the 2,400 to the 4,470 meters above sea level, as shown in this graph, the elemental composition is about the same. What changes is what you can find the soluble fraction, and some of the measurements that we've done as shown here. We have a marked pH gradient where alkaline soils at the bottom of the transect and acidic soils at the top of the transect. And also this influences the availability of nutrients. As shown here, we have a graph showing nitrogen in green, phosphorus in orange, and potassium in blue. So potassium, for example, is not quite rather limiting, as shown here in reference to the 100% line, and it's particularly abundant towards the lower end of the transect. Phosphorus is limiting in about half of the transect, and then it's okay towards the top of the mountains. But nitrogen, for example, is quite limiting throughout the entire transect, and in fact is about 5 times lower than the global mean nitrogen content for comparable deserts. So the combination of environmental parameters from which water, as I mentioned, increases toward the top, and temperature, which decreases toward the top, are the most probably the most important. Also, the lack of nitrogen or sufficient nitrogen, and also high radiation, defines 3 ecosystems: the steppe, which is dominated by perennial grasses, as you can see here in this picture, and the puna, which is the most diverse ecosystem in this transect, which is dominated by shrubs and subshrubs, and the prepuna, which is the most extreme environment near the edge of the desert, of the absolute desert, which is dominated by annual plants and succulents. So one of the first questions that we had when we started this project is to actually know what is the diversity here. And this is not such a simple question when we look at an extreme environment such as this, because many plants actually escape the most extreme parts or the most extreme seasons, and you cannot see them, for example, if you go surveys, unless you do these very frequently and for a number of years, which is actually quite hard to do. So we combine survey work for, in this case, what I'm showing here is for 8 years, but also metagenomic analysis of soil DNA, pollen rain analysis, and soil seed banks to really try to have a sense for what's the diversity in terms of plant species in these 3 ecosystems. And so what I'm showing here is a summary of this. I don't mean to go over all of these, but just to say that the transect contains 92 taxa that we have been able to identify, and 77 plant species that we were able to identify. It's dominated by angiosperms. We only found 1 gymnosperm species with Ephedra bryoana. With this information also, we've been able to reconstruct the plant diversity landscape over the years, even going back to 27,500 years ago, using fossil records. And I'm just showing this just as I guess a teaser, but if you're more interested, you can look at this reconstruction over millennial timescales in this global change biology work. But for the purpose of this talk, using this information about diversity, we selected 32 what we call ecologically relevant species for genomic analysis, primarily using 3 criteria: coverage, so how much of the ground these species cover; persistence, that is how many times we find these species when we go. So obviously, if the plant is always present and it has a high coverage, it's probably very important in ecological terms, and also the distribution, so how much of the transect it covers. And these are the 32 species that we are gonna analyze in more detail in a minute, from different plant families. As I said, most of them angiosperm, except this gymnosperm. They have different photosynthesis types, C3 and C4, different life forms, and some of them are endemic species. And what's shown here is where these species are located, and the intensity of the blue in this case shows the persistence over 9 years. Again, this is the elevation, and these are the 3 ecosystems that we analyzed. These are pictures just to show you the life habits of this plant, to have a sense for what we have sequenced. So we have poaceae, we have solanaceae, which are probably important if we think about relevant crop species. Fabaceous as well, but other families are represented: Asteresis, Amaranthus, Apiaceous, etc. So for all of these, we did Illumina sequencing, parent sequencing of the transcriptome. I'm not going to go over details of the bioinformatic pipeline, but basically we did de novo transcriptome assembly, protein prediction for functional annotation in order to try to understand what's unique about this plant species that may explain the ability that these plants have to survive in Atacama. And I like to always show this picture because just to give a sense for the scope of this work, because this is an image that I took in one of the stations in the transect. And really, we have sequence data for all of the species that you can actually see, which are the dominant species, as I said, in terms of the coverage, distribution, and persistence in this ecosystem. So now the question is, what are the features that these Atacama species have that allow them to survive? I should also say that we've complemented the sequencing data of the plants with the microbiome associated to the plants. And I'm just going to go very quickly over this just to show some highlights. We did, in collaboration with Mauricio Gonzalez, a colleague in Chile, and Jonathan Maldonado, a postdoctoral researcher, sequenced the microbiome sequence of bare soil near this species, particular species, and also the microbiome and the root zone of these species. And as you can see, there is a difference in terms of the bacterial taxonomy. So, for example, the root zone has more Pertobacteria than the bare soil. There's also more Bacteroidetes. And associated to this change in the microbial composition, we have also differences in the putative functions that are associated to these taxonomical units. And in particular, one thing that I would like to highlight here is that we have predicted functions related to nitrogen fixation and nitrification. And when we validated these predictions using sequencing of the NIF-H marker, which indicates presence of at least the potential for nitrogen fixation, we have an enrichment in the root zone of these plants as compared to the bare soil. So this suggests that the differential selection of microbes associated to Atacama plants may also be contributing to survival, in particular as it relates to nitrogen fixation in these low nitrogen soils. Now, if you now look at the plants species, we have over 880,000 total protein coding genes identified, more than 486,000 plant protein coding genes, many new protein coding genes. And the work that I'll show now, it was done in collaboration with Gloria Carusi at the Center for Genomics and Systems Biology, New York University. And here Kranti played an important role in the first part, and now Gil also has made important contributions for the positive selection analysis that I'll describe. So in order to understand what are the unique features of the Atacama plant species, we compare the sequences that we had for Atacama with 32 sister species. These are phylogenetically related species to the Atacama species, but they don't live in Atacama. They live, in some cases, in mesic environments. And we also included 6 model species just to bring annotation, as these are species where we have more information. So with these 70 species, 32 Atacama species plus the 32 sister species and the 6 reference genomes, the first thing we did was to build a phylogenomic tree. And the pipeline that was used for that is highlighted here. So basically, in the first part, in this first row, what we did was to define orthologos for all these species, starting with gene clustering, alignments, building phylogenetic trees for each gene family, and from these phylogenetic trees, define orthologs for all of the species that we have, these 7 species. With these orthologs, we built a phylogenetic matrix that contains the protein sequences on each row for each of the species. And with this complete phylogenetic matrix, we build a total evidence tree as indicated here. And it's this tree that we have used to try to understand what are the features, in this case, protein sequences that distinguish Atacama species from the sister species. And for example, as I'll show, try to identify evidence for evolutionary evidence for selection, for example, for positive selection. And then by looking at the genes, then we can look at biological processes of using gene ontology categories, for example. So we have 32,292 families, 15,972 orthologs, 8.5 million amino acids approximately. So this was a big job in terms of the computational power needed to be able to build this tree. So I'm just flying through this, but this took quite a bit of time, and Kranti was instrumental to get this done. So just the runtime, once the pipeline was optimized and ready to run, took 1.5 months to complete. So this represents 1.7 million genes, and as I said, more than 8,500,000 amino acid characters for this set of ortholog genes. So this is the phylogenetic tree that we built. And in here in bold are the Atacama species, and in normal font, on regular font, you can see the sister species and also some of the model species. Okay, so here Gil used two approaches to try to identify evidence for positive selection. Tree-dependent DNDS modeling using CodeML, using both species-independent adaptation and also ancestral adaptation. So with the species-independent adaptation model, we found 152 positive selected genes, and with the ancestral adaptation, 117 positive selected genes. Gil also did 3 independent DNDS calculations using SNP genes, and here we're basically comparing groups, so 2 groups of species. And the comparisons are indicated here. So we did drought adaptation comparison, where we took drought adapted sister and mesic sister species, or drought adapted and the Atacama species with the mesic. And also we had nitrogen adaptation comparisons, where we compared the low nitrogen soils, so species that grow on low nitrogen in Atacama versus drought adapted species that grow under high-end soils, or mesic species. And here are the numbers of 5 PSGs for this comparison, 6, 77, and 15 in the last comparison, for a total of 85 positive selected genes identified using this strategy. So overall, we found 265 gene families under positive selection in Atacama using all these approaches that I just mentioned. And this graph shows presence or absence of these genes in all of the species. So here we have the species at the top, the Atacama are in red, the sisters are in black, sorry. And the plant families are color-coded again. So as you can see from these presence-absent graphs, some gene families are clade specific. So for example, here we have a big clade that had from the poaceae, from the grasses. Here we have some from solanaceae or favaceae, but some also gene families have evidence of positive selection across all the species that we looked at. So these color codes indicate what type of analysis provided evidence for the positive selection. In some cases they overlap, in some cases come from one of the analysis. And here in the last column we have top expressed genes. So that means that these genes are not only under positive selection, but they're also very highly expressed in the Atacama species, which might indicate that they're not only under selection at the protein structure level, but they may also be under selection at the regulatory level. If you look at the PFAM domain, so these are annotated domains in proteins that have functions, we have about 67% of the proteins in the positive selected set that have a PFAM domain. And interestingly, for all of these that have a PFAM domain, the majority, 64% of them, we have the site with the positive selection within the PFAM domain. This doesn't demonstrate necessarily that this it's relevant, but it certainly indicates that the positive selection is occurring in a functional domain, and this may be important for the function of the protein and therefore for the adaptive capability or possibility of the species that contains it. These graphs show the different PFAM domains that we found. Some of them are overrepresented, which are indicated with these blue stars, like DNA J domains, so related to our previous talk. These are domains are common in chaperone proteins. But we found a number of protein domains, such as protein kinases, acid transferases, also zinc fingers, which are found in different types of proteins, for example, also in transcription factors. If we now look at the gene ontology annotations, we found a number of processes that appear to be under positive selection because the genes that are we identified have common functional annotations, and these are highlighted here. So response to stimulus, for example, biological regulation, developmental processes, cellular component organization, a big metabolic process component, and also localization. The color here indicates the number of positive selected genes that are annotated to this process. So for example, here we have a number of positive selected genes that are annotated to cellular nitrogen compound by a synthetic process, and this is also overrepresented. So this would indicate that this is a biological process that is important for adaptation, and it also makes sense related to the low nitrogen environment that the plants experience here. To further understand how these PSGs may have contributed to the Atacama species adaptation, we reviewed the literature for phenotypic and functional characterization of these positive selected genes based on the closest sequences from the model plant Arabidopsis thaliana, and we found phenotypic evidence for 59 of these PSGs. And I'm gonna just summarize this analysis, this extensive analysis of the literature. So we have positive selected genes that facilitate coping with high radiation, also involved in photosynthesis, which are critical for chloroplast development and movement, for the integrity of the photosynthetic apparatus, for example. Also, we found genes involved in modulating flowering time, which may be particularly critical in this environment because, as I mentioned in the beginning, water comes in a very short period of time during the year, and so plants must grow, in particular in case of the animals, they must complete their life cycle really quick, flower, and proceed to the next generation. And also in below ground processes, we found genes involved in biotic interactions, genes involved in enhancement of root hair growth, and also relevant to low water capacity and some of the limitation in terms of nutrient availability. We find genes involved in optimizing water capture and also enhanced nutrient use efficiency. In some cases, the positive selection is within the domain, which is indicated with this blue bar with the red dot. Many of these are also highly expressed in at least 2 of the Atacama species. And these dotted lines indicate stress tolerance phenotype that has been characterized in Arabidopsis either by mutants or overexpressors. So with that, I would like to finish. So I hope that I'll show you how we're using big data sets, in this case, transcriptome data set from Atacama species, integrated bioinformatics in phylogenetic models to provide hypotheses regarding new genes and new processes that are important for survival in Atacama. I think some of them, as I showed, have been described in model organisms, but there is about 20% of them, but many of them are still not studied. And so I think this is really a veritable gold mine of new functions that may be relevant for example, crop improvement to tolerate increasing desertification produced by global climate change. Just to acknowledge that the people that did the work, so many people have participated in this project over the years. Most importantly, Gil, Viviana, and that are here, and Soledad, in the instrumental in some of the analysis and getting this in good shape for publication, which we are still in the process of. But then a lot of people, Daniela, Carol, Alejandro, and Tomas, for example, helped with the bioinformatic analysis, as I mentioned, Kranti as well, Jonathan with the microbiome analysis, etc. Gabriela in the diversity. This project was initially funded by the Center for Genome Regulation, and some of the initial exploratory work we did also under Howard Hughes funding. But we had a grant with Gloria from the Department of Energy, which allow us to complete this study and also start a similar study in the California desert, which is also coming in the future. Here are some pictures from the Atacama field trips over the years. And again, just mention the huge role of Claudio to be able to start this project. And over the years, Mauricio, that's done the work in the microbiome, and the Evanet team, which I already mentioned, Gloria, Cranti, and Gil, has been very important, but also Dennis Stevenson at the New York Botanical Garden, Rob Desal, the American Museum of Natural History for all the phylogenomics, Jean-Michel Lanet and Heidi Kepler at Wisconsin for characterization of the function of these genes, and Rick McCombie in sequencing. And with that, I'd like to finish. And thank you for your attention. I'll be happy to answer any questions.
Thank you very much, Rodrigo, for the excellent presentation. So we have time for 2 questions. So the first one are asking if you have an idea about the polyploidization events in the species that were analyzed, and how that could affect the selection analysis.
We haven't done analysis of polyploidization at the moment. We haven't looked at genomes. Just based on the assemblies, we can tell that there are probably some species that are polyploidy and others are not. But we haven't gone in too much depth in that sense. All this analysis we've done with pools of plants, so I think this shouldn't be too much of an issue, as we are probably picking up the most abundant genes in the population, or even within a given individual.
Okay, final question. Do you find any indication of adaptation to high UV radiation and/or DNA damage?
Yes, I think we do have some evidence for genes that may be important for those traits in particular, as I mentioned in one of the last images. Yeah.
I should say that there are like the photolyase gene,
yeah,
which is known for UV radiation repairing damage, and all the ROS genes that are there.
Right.
Okay, thank you very much, Rodrigo, and thank you very much, Elizabeth, for your wonderful talks today. I would like to also thank the audience for the great questions and the discussion. And with that, I would hand over to Katie.
Yeah, also thank you, Jill, for the excellent moderation. This has been a really great discussion. Thank you all for joining, and I hope that you can also join us on our next Plantae Presents webinar. You can find those on our Plantae blog, and I hope to see you there.